which means you guys probably shouldn't share anything that's incriminating at this point. And okay, oh wait, that's the, there you go. Are you guys seeing the like right picture thing now? Is it okay? That's good. So anyway, thanks everybody for showing up, and that for those of you who are watching it afterwards. But hopefully, even for some of you that like have been participating in this before, up until now, like you could learn something from it. We want to kind of talk about the chemistry of cytochrome P450 and why that matters, obviously, for our project. But let's see. Hold on. Got a text saying they can't find the link. Yes. Dang it. Oh, hi there, Shaylee. Can anybody see the details of this meeting to see if Heidi's included?
It looks like she's not tagged on the email.
Oof.
I don't think Tracy is either. I think it's just students.
Okay. All right. I think I resent the thing. So, oh, hey, Shaylee, since you're here, do you want to kind of introduce yourself to, I think Sally and Jocelyn are here. I think we talked about your name. If you had to pick, would you identify as a biologist or a chemist? And your choice of superpower or something like that.
Yeah, so I'm Shaylee. I graduated from Waynesburg, but I worked on the NSF project for like 2 years while I was there, and I'll probably be there. I'll be there a little bit in the summer. I'd probably identify myself as a chemist, even though I still do like biology, but still probably chemistry. As for a superpower, probably fly.
That's cool.
Yeah.
Yeah, I hope you have a good insurance policy for that. But hey Heidi, if you, I don't know if you are set up to talk, but like, do you want to introduce yourself? Because we have some new people here today, too, Sally and Jocelyn. Before I get going on the thing.
Let's see here. There we go. Oh, this one.
Oh, now we can hear you.
There we go. Can you see me? Okay. Hello. Yeah, I'm Heidi. So I'm also working with Talk and Tracy on the project with you guys this summer. I apologize, I missed the last the first meeting that you guys had. So yeah, I don't know what else you want me to talk about.
What's your superpower of choice, and do you identify as a biologist or a chemist?
Yeah, I probably would say fly also as a superpower, and yeah, definitely chemist.
Cool. All right. Thank you, Heidi. And hey, everybody, like as I go through this stuff, please, like, interrupt. Feel free to interrupt and ask questions or complaint or something. Okay. Let's see. All right. Cool. All right. Nice. So like I'm always kind of in these like trying to get back to the level of like maybe Gen Chem, so some of you guys could, who haven't had OChem and whatnot, could appreciate this. But I'm sure you guys talked about like catalysis of a reaction, how that lowers the activation energy of a reaction. And so that like a reaction that maybe normally has way too high of an activation energy, so like it would never happen in Earth's time scale type of a reaction, could still happen with the help of a catalyst. And usually the actual mechanistic pathway looks complex like this in real life. Like oftentimes you go through more than one intermediate molecule in between the reactant and the products. And because you go through this completely different pathway that requires less energy, that's why catalyzed reaction, might have a lot lower activation energy and can happen at a reasonable time. And each of these welds would be an intermediate in the mechanism. But you could do the same thing with an enzyme being the catalyst, which is like a protein being a catalyst. Where if you have in that context, we tend to say, well, the starting material substrate for whatever reason. But so the enzyme substrate complex is sort of the equivalent to intermediate molecule along this reaction pathway. And again, it lowers the activation energy, so things happen. The enzyme that we're working with, Cytochrome P450, is an catalyst enzyme. To be familiar with the chemistry, this is kind of the good way to think about it. It's sort of the nature's tool to make molecules polar, because you never know when you're exposed to accidentally some chemicals that we might call xenobiotic, because they don't really belong in your metabolism or in your physiology, but you still need to be able to survive that event, right, as a living organism. So this would be true for pretty much anything like bacteria, plants, animals. And some chemicals you might get exposed to, like, I don't know, like a crude oil or something, is so non-polar that once it's in your system, it will be impossible to get it out through normal means, because, like we are all um, water-based organisms. So like it's usually easier if whatever you're trying to get rid of from your system is soluble in water. And cytochrome P450 often adds functional group that makes the molecule more polar. Besides the dealing with xenobiotics, cytochrome P450 is involved, at least in humans and, well, in a lot of organisms, a lot of normal physiological metabolism, like synthesis of a lot of the steroidal-based hormones that we have. All the circled steps are actually cytochrome P450 enzymes in human steroidal biogenesis. Okay. Some of these, like you notice, a lot of these you notice, like it puts a hydroxyl group or alcohol group right there in that step. Sometimes it's to oxidize a certain functional group, like in a little more interesting way. Like here, it involves like a oxidative um, deacetylation reaction. and whatnot. But those are all cytochrome P450 chemistry. Other things that you might see, especially in humans, that is medically relevant might be like a lot of these icosanoid compounds that are involved in usually sort of bad things like inflammation, dealing with inflammation and or causing inflammation and stuff like that, um, are derived from polyunsaturated fatty acids through the a lot of the steps are catalyzed by specific cytochrome P450, which by the way, all of these come from uh, all these icosanoids are compounds that are always oxidized at the 6th carbon from the end, or omega-6 position. And that's why you might have heard, like, you know, some people talk about like the balance between omega-6 and omega-3 fatty acids. Omega-6 fatty acids have a unsaturation or double bond at 6th carbon from the end. Omega-3 has a double bond at the 3rd carbon from the end. Omega-6 tends to lead to these inflammation-causing icosanoids, which we modern humans, I think, have almost too much of. But anyway, going back to that idea of xenobiotic metabolism, that's something that, like, if you're a doctor or a medicinal chemist or a pharmacist, you have to think about a lot of. There's 2 phases of metabolism, at least in human physiology, to get rid of xenobiotics or compounds that normally don't belong in our body, like drugs. The phase 1 is usually done by cytochrome P450 to make metabolites that are, like we said, more polar. So those compounds usually get eliminated in your urine because now it's soluble in water. In phase 2, it's called conjugation. You add some other functional groups or molecules that your body's kind of familiar with, like sugar or amino acids, and then you make stable adducts that, if it's nonpolar at that point, it comes out in your stool. If it's polar at that point, enough to be dissolved in water, then it might come out in your urine. And this is kind of showing the same thing, but showing the chemistry of it. But a lot of this has to do with adding hydroxyl groups. But the main thing that's pertinent to our project is cytochrome P450 in general, because they're designed to deal with xenobiotics. They tend to accept a wide range of substrates. So that's kind of why we're interested in the enzyme. This is one example of dealing with a xenobiotic. Like, you might have heard of codeine. This is a specific cytochrome P450 in human. Is converted to morphine, so it's considered a pro-drug. And then this happens by adding alcohol on that methyl group, which then this, if you've taken organic chemistry, you might recognize that as a hemiacetal functional group that's easily hydrolyzed, and so the aldehyde comes off, and then that becomes morphine. So anyway, and this is of course not like a normal compound that you find in your body, but your cytochrome P450 deals with it because it has a broad substrate scope. So so far the highlights, if you want to reduce all this down to like 3 points, the broad substrate scope is important, and cytochrome P450 is considered a monooxygenase, typically puts like a hydroxyl alcohol group on it. And we'll look at this a bit more, but that is done by single electron reaction mechanism, which is kind of advantageous because that's typo, sorry, it should say radical. Radical intermediates, they are very high in energy. So if you can produce something like that, then a lot of reaction mechanism pathways are possible from that high energy intermediate. And so that's why like some reaction products that are often difficult to obtain, it could be obtained from a mechanism like that. And as far as the structure of cytochrome P450 protein, they often look like this. The one thing that is conserved among all cytochrome P450 that is this cysteine residue, which when we say conserved in this context, it just means like all the cytochrome P450 proteins have a certain amino acid residues in common, and one of those is the cysteine residue. It's hard to see, but there's a cysteine like right there. And if you've had like biochem or something, cysteine is that one amino acid with a thiol or sulfur group that is actually covalently bonded to the iron of the heme B molecule, which looks like this. And this is where sort of the chemistry happens in cytochrome P450. It's all thanks to this iron chemistry. Now, I talked a lot about cytochrome P450s in humans, but these are all membrane bound, whereas in bacteria, usually they're soluble globular type of protein. And so that's why like we're interested in the bacterial cytochrome P450 because we want it to be soluble in aqueous reaction mixture. And so anyway, that's sort of part of the background, but I should probably have asked at this point, like, does anybody have any questions or anything that like I didn't explain very well, or maybe even Heidi want to jump in and correct or add something? I don't know.
The only thing is one of the main goals that I think I'm supposed to work on this summer is if you go back to the previous slide you had there.
Yeah.
We're trying to isolate, right, I'm going to try to purify the protein.
Right. Yeah.
Right. Yeah. So it's not membrane associated, which in our procedure we don't have to deal with that. So that is good news.
Right.
For
Yeah.
bacteria. Yeah.
Yeah. Although now that you mention it, some of the new cytochrome P450s we bought from Adgene, like BM3 and CYP119. I know that they're bacterial origin, so I assume they're soluble proteins, but I guess I haven't specifically seen in the literature or something that they definitely are. So it's probably good to double check, but I'm pretty sure they're not membrane associated.
Okay.
Either, but yeah.
Just so they combine to the resin. That's the main thing, right? That we have to.
Right.
Yeah.
Yeah. So maybe we should double check. I'll make a note of that here. But thanks for pointing that out. So the heme molecule is sort of crucial for the catalytic activity. So like even if we're making that cytochrome P450 protein, if there's no heme, the chemistry is not going to work. And so it's kind of important for us to know like where that comes from. And this is one biosynthesis pathway that's interestingly like pretty similar in like all the organisms, like whether you're talking about like bacteria or humans, because we all use similar heme molecules or sometimes the same, like heme B for peroxidase or monooxygenase type of enzymes and or proteins. And then it all starts with this compound 5-aminolevulinic acid. That's sort of, to use the biochem lingo, that's the committed step, meaning that like prior to this step, like all these other metabolites leading to this could become other things, but once it becomes 5-aminolevulinic acid, this is not destined to become anything else other than heme. So there's quite a few steps after the committed step, and then then you make your heme molecule. But all of that to say, uh, having some knowledge of this background, I think is kind of helpful, even when you just do your lab work in the lab working on this project, because this explains why we have to add this stuff, add this stuff, 5-aminolevulinic acid. That looks good. And as far as the actual chemistry of cytochrome P450 goes like this, there's roughly 8 steps to this. These 8 steps that I'm going to maybe walk you through real quick, step by step. So the first step is when the substrate binds to the whole protein. And this may not happen exactly like right where the heme is, but it's usually somewhere kind of close by. And that changes the conformational change, or that changes the shape of the protein, so that like iron is willing to let go of that water normal ligand. And sometimes that recruits another associated partner protein like cytochrome P450 reductase that uses cofactors like NADPH or NADH to reduce the iron core metal from 3 to 2. And then so that's the second step. But something like this usually has to happen so that iron, because iron 3 is not very reactive with oxygen, but iron 2 is, because it can oxidize or reduce, excuse me, the molecular oxygen, which I think a lot of the Gen Chem type material I've seen when people learn how to draw loose structure, I feel like some textbooks or something showed the oxygen structure to be like this double bond thing, but that's actually not correct. That would be called singlet oxygen. Oxygen looks more like this, like a diradical thing. That gets reduced by iron, and then you get to a structure that looks like this. And then in the fourth step, this needs to be further reduced, and unless there's like a peroxide shunt mechanism from this, sometimes that happens with the cytochrome P450 reactions, but usually this gets reduced to the peroxide, like this is like a superoxide, I think, oxidation state. And this is often done by another partner redox proteins, like oftentimes it's ferredoxin. If you've been working on this project, you might have heard, I'm sure you've heard of the protein in the marine bacteria that we work from called BMP9, and that's a type of ferredoxin. Just a trivia fact, but our BMP9 cytochrome P450 is actually known to be able to work with sort of non-cognate ferredoxin or unrelated ferredoxins. So it's theoretically possible, if you have forgotten to add BMP9, that the cytochrome P450 still could work with a native ferredoxin from E. coli, the host bacteria, but probably not as well. And no one knows how much ferredoxin our host E. coli actually produces. So we still need to add it. And the only thing that it complicates is hopefully we are actually producing this BMP9, because even if we are not, it might kind of work, the whole reaction. So we might not notice it unless we specifically check for this, which I think Haley's been working on something like this a bit, just to visualize BMP9 on a page gel to see if we actually are making it. But anyway, that's kind of a side note. But ferredoxin is an interesting protein that has this like 4-membered ring with 2 iron metals in it. And so that's this abbreviation here caught up in this ferredoxin protein that's held in place by the 4 cysteine residues. Or cysteine, remember, is that weird amino acid with sulfur in it. And then often, so these iron core part of it with in the ferredoxin either gets reduced or oxidized, is in an oxidized or reduced oxidation state, and that's what um, provides this extra electron needed for that reduction. And then this in turn often needs to be um, reduced or oxidized depending on the direction of the reaction by a partner reductase or oxidoreductase enzyme. Like in our case, there is a ferredoxin reductase called BMP10. And usually ferredoxin reductase requires an FAD cofactor, excuse me, that as well as some sort of an NAD-based cofactor, whether it's NADP or NAD. This, well, NADPH is often the ultimate like reducing agents, electron source that like reduces the FAD, and then FAD reduces the ferredoxin, BMP9, and so forth. And so that's part of the reason why, like, if you've been working on this project, or if you are going to start working on the project and have read the protocols or something, you might have seen like some of these things mentioned as essential part of the experiment, and it's because of that. And the reason why the cytochrome P450 sort of wants to reduce it to the peroxide oxidation state with a formal negative charge here is once that gets protonated, like twice, that oxygen bit could leave as a neutral water, which would further oxidize this, which would oxidize this the other oxygen so badly. But you know, if you've had even Gen Chem, like, you know, oxygen is very electronegative, wants to hold on to its electrons quite a bit. So this oxygen is not going to stand for that. So it'll just take electrons from the iron, which makes it into this like very unnatural and unusual iron +4 oxidation state. Iron is only comfortable being +2 or 3. So that's a super oxidized state, and that's what sort of makes this whole thing possible. In the next step is this is so super oxidized that it wants to be reduced. It wants to gain an electron badly. And one way it does that is by abstracting a hydrogen radical or atom from just about any substrate that's close by, like that this generic substrate that might have attached to this protein kind of nearby. And so that would, of course, get you a radical species because you just took out H dot. And then this often reacts with this formerly a hydroxyl radical attached to iron 4. And if it takes away hydroxyl radical to make the covalent bond, this is now formally stable alcohol molecule that could still be bound to iron as a ligand. And once that product is released from the enzyme, replaced by water molecule, you know, this can go through the whole catalytic cycle again. And that's sort of how that reaction happens. And we said that NADH or NADPH is the ultimate electron source or reducing agent source, which this is what NAD looks like. NADH would be one with another hydrogen here. Looks like this. Now, NADH, if you've ever had to shop for a chemical, is actually a super expensive chemical. And to provide that in a stoichiometric amount for really any size reaction is just too cost prohibitive. And so most of the time when people use Cytochrome P450 chemistry, you provide the stoichiometric amount of NADH or NADPH by having a coupled enzyme like a phosphide dehydrogenase that could take a cheap reducing agent like phosphite and then reduce NAD plus continually to continually make NADH. And so that's why oops, you know, if you've been working on this project, you've probably heard of that enzyme PTDH, but this is why you need it. So far, just to kind of summarize what I've said here, you know, for cytochrome P450 chemistry to work, you need redox partners like ferredoxin and ferredoxin reductase. Usually the ferredoxin reductase needs stoichiometric amount of nicotinamide-based cofactor like NADH or NADPH. And those are expensive, so you typically continually regenerate this with the use of some sort of coupled oxidoreductase like PTDH that can take cheap reducing agent like phosphite. And then we need the heme as a cofactor for the cytochrome P450 itself, which is biosynthesized from 5-aminolevulinic acid and iron. And a lot of organisms share that biosynthesis pathway in common, and that's why usually just by providing 5-aminolevulinic acid, you can sort of trick something like our E. coli host bacteria to make heme. And 5-aminolevulinic acid is after the committed step, so that tends to usually stimulate the downstream biosynthesis down to heme. But I just have a few more slides here, I think. But does anybody have any questions or comments or complaints or corrections or anything like that? Or any addition? Luckily today I'm not losing my voice as bad as last time, so I think I can make it through the end. Praise the Lord. But so I mentioned that like substrate often like attaches or is attracted to cytochrome P450 a little bit far away from the heme binding site or the heme itself. So here's your heme. These are some known substrate recognition sequences. And you might think, well, gee, that's a lot. Why so many? But remember that cytochrome P450 is kind of designed to, oh man, accept a wide range of substrates. And so especially because like sometimes they're designed to specifically deal with xenobiotic compounds, especially if we're talking about humans in the liver. Yeah, they can accept a wide range of substrates. Now, that's not always the case for some of the bacterial ones, but they often do accept more than one substrate. And that's why we think we could take advantage of such an enzyme to sort of trick it into making a compound that we want that would be useful for medicinal purposes. So that's kind of the gist of our whole project. Specifically, we're kind of interested in developing a whole new biocatalytic platform. This is kind of like a bigger picture beyond just this project per se, but is in organic synthesis, we often do something like this, transition metal catalyzed coupling reactions. And those are reactions that like couple together like big chunks of molecules that have a specific functional groups, and then you can combine them together. Like Suzuki reaction is kind of a bread and butter in organic synthesis. But when it comes to like a carbon, I mean, a heteroatom to carbon bond coupling, there's like Ullmann coupling, Buchwald coupling, those are famous and often used, but those things tend to require like really high, harsh temperatures and conditions. And especially when these aromatic rings are heteroaromatic rings, as in they contain like nitrogen and whatnot, and they tend to be very labile or unstable. And so if you have to use these harsh conditions to make such coupling products by organic synthesis, that often doesn't work for those sensitive situations. And so what we want to do is to provide sort of make or invent biocatalyst that's engineered the cytochrome P450 that could do these coupling type of a reaction that normally would not be possible with conventional coupling technology and technologies in organic synthesis. So that's hopefully a good background knowledge in cytochrome P450 chemistry. But any questions or complaints? That's pretty much it, all I've got for you. And where we're going with this next meeting, and again, I apologize, it's back to back, that it'll be tomorrow, is how we might, in practice, do this engineering of this protein. And so it's more of a biology side of things we want to talk about, but any questions or comments or anything like that? Or if it even gives you some like side project or like ideas to do during the summer, we could talk about that too. Anything?
I just connected a lot of dots in my head.
Mm.
That side of knowledge really helped understand like why we add things.
That's good. I'm glad to hear that. Although, well, actually, maybe Haley, you might be a good person to ask this. Like, without having worked on this at all, like if you can imagine, like you like haven't worked on this at all as a freshman, like maybe some of these things make sense to you because you've been working on the projects, but like, would this have made sense to you if you've never worked on this project? And I gave you this as a lecture.
I think that it like makes sense in the way that like you're absorbing the knowledge, but a lot of it will piece together once you're in the lab and you're working with the pieces of it, because you're like, oh, we add this during this step, versus like, oh, I just know what this is called. Like, I mean, I did it in the reverse order where I was just adding them, and now I'm like, oh, now I know what it's for. So it might seem like really heavy and really like subjective, but once you start working with them, you start piecing things together. And personally, like when I started working on the project, I would always ask questions. We heavily encourage it because there is a lot that you're not going to understand at first. But like I would always ask Shaylee, I'm like, why do we do this? What am I doing? It's like it just, you start to slowly piece it together. So if you're not understanding now because you haven't worked on it, just know that over time it does make more sense.
That's good. Thanks for sharing that. I especially want to hear some questions or something from our high school students. If you're not too intimidated to ask something, like I bet there's something that.
I mean, I think I understand a lot
Yeah.
of the concepts because I've taken
Yeah.
biology and chemistry.
Good for you.
at college levels, but I think it'd be good to like see it in practice and like actually doing these things to better understand, at least for me.
Yeah. Anything that kind of went over your head that like you wish I'd explain a little differently or explain it again or something like that?
I think I have a good idea generally of the concepts that you're going over here. So
Okay. All right. Yeah. Thanks for sharing that. So I think that's pretty much it. I can't remember if I mentioned this to all of you at the beginning today, but so I'm really sorry about like such a frequent change of plans and schedules. But tomorrow, so I just had, maybe you don't need to know all this background, but I just had a meeting today with our, separate from this, like I have a project working with biotech, like pharma company. It actually technically wasn't my fault, but like another partner we are trying to work with, we have to have a meeting tomorrow, but we just found out that they messed up the time and the meeting was supposed to be today, but like they were not for some reason available and they can only meet with us tomorrow at, shoot, I forgot, 5 o'clock our time. So I think our schedule, our meeting, we had scheduled it for 4:30, but if you don't mind, I'm moved it to 4 o'clock, just because I need to be ready for that. I need to be available for that 5 o'clock meeting, and I just couldn't move their schedule. Like it's a people from Mexico and Alaska and Chicago all together in one meeting, so it's just too much to coordinate. So I apologize. Um, so again, I hope you can make it. If you can't, I'll record it and then uh, I think what I'm gonna do is I'm gonna upload it to a YouTube account and then share that link with you all so that you can watch it if you miss it. Or speaking of like things making more sense after you are in the lab, like some of this maybe it might be useful for you to refer back to after you're in the lab or something as well. So anyway, any other questions or
did you
anything?
say 4 o'clock tomorrow?
Yeah,
Yeah, okay, I think that works. I might be a little late if that's okay.
yeah, that's okay. Yeah, and any other questions or comments or anything? It doesn't have to be even about this sort of topic. If anything has come up, like you know, anything logistical or anything like that.
I did want to offer for the high schoolers, if anyone has any questions about like, dorm life, laundry situations, like I can
Yeah.
give them my phone number and they can reach out to me and I can answer questions about like, living arrangements, things like that. If anyone wants to stay, I can give you my phone number.
Cool.
Yeah, I'd be interested in that, just especially with it coming sooner. Just
Maybe you can
Me as
put it
well.
in the chat or something.
Okay, I'll put it in the
Yeah.
chat.
Yeah. Yeah, that's great. Thanks, Haley. Also, did you want to share that you have a license now?
I do. I did get my license, so I will have a car if anyone needs like a trip to the grocery store or something. Just let me know.
Great.
yeah, I also just got my license today, actually.
Oh wow. Oh, today. Well, congratulations. Nice. Do you think you'll be driving here, Jocelyn, or?
No, because I live in Florida, so I wouldn't be able to drive up there. I'm gonna fly there, so.
Yeah, I see. Yeah, that's cool. All right. Anything else from anybody? By the way, Shaylee, that's a nice yard you have or something. Background looks nice.
Thanks. It's my aunt's
Yeah.
house.
Oh, it's your aunt's house. Oh,
Yeah.
cool. Well, I'll say hi to your aunt just in case she can hear me.
It's as she can, but it's actually a golf course.
Like, whoa. Wait. Your aunt
Yeah.
owns the golf course.
Well, she doesn't own the golf course, but it's just in her backyard.
Wow. So isn't that kind of dangerous? Like sometimes maybe the golf balls come and break your window or something. That's interesting.
Yeah.
But that's nice. Cool. All right. If there's nothing else from anybody, I'm gonna go make dinner and stuff. But thank you guys for attending. Some of you, I guess I'll see you tomorrow, and then, or if not, in the next meeting in a week or so. So, all right. See you guys. Bye bye.